As we continue on talking about genetic variation, not only is genetic variation important within a population, but it's also important between populations. A lot of times you have geographic variation. So you have variation between populations that are located in slightly different places. One good example of that is if we talk about giraffes. Giraffes do have some differences in regard to their coat and the pattern of their coat. And it really is very different depending on where they're located in Africa. So the different coat colors or coat patterns are located in different places. So that would be what we would call geographic variation. That's something that we can visually tell when we look at these different giraffe populations. Some variation, though, that you have between populations is not as obvious. So it can very well be something that is just in the DNA, and you wouldn't know unless you actually went and did a karyotype or analyzed DNA sequence. And here's a good example of that from your textbook. These are house mice, and these are in two different locations, and over time some of the chromosomes have been fused together and some have been deleted, and that hasn't really changed the appearance of the mice at all whatsoever. So visually you can't tell the difference between them, and that's because they still have the same genes there, they still have the same number of genes, it's just that they're arranged on the chromosomes a little bit differently than before. So both of these are examples of what we would call geographic variation among or between populations of a species. Now here's another example yet again, and this represents what we would call a cline. So a cline is basically a graduated difference in phenotype along some type of geographic difference or range. So in this case, we're looking at altitude. We have this same plant species growing at different altitudes, and depending on the altitude, we see a difference in height. So the height seems to be related to the altitude level. The higher the level, the shorter the plants, the lower the altitude, the taller the plants. Sometimes you see this with a temperature range. You might have some variation there. So they might have a different thickness of fur if they are located at colder temperatures compared to more warm temperatures. So a cline is just another example of variation among populations of a species. And this probably does have something to do with natural selection. It's probably being selected for naturally. And that's why this difference in phenotypes is being maintained in the population. So now the big question is, where does genetic variation come from? So if it is so important, how do we get this genetic variation that we're seeing? How do we end up with 14% gene diversity in humans or 0.1% nucleotide diversity in humans? So one of the biggest ways that we get genetic variation is from sexual reproduction. Sexual reproduction, if you'll recall, is basically we're taking an egg and a sperm, we're fusing those together, and then we end up getting what we call a zygote. Now, that egg and sperm, recall how those are actually produced. Those are produced by the process of meiosis. And meiosis itself contributes a lot of genetic variation and variability to the process of sexual reproduction. So in meiosis, you do have crossing over that takes place. Hopefully that is familiar to you. We do have also independent assortment that takes place. We are reducing the chromosome number. Okay, so since we're reducing the chromosome number, we're not going to have all of the content there that was present in the parents that were producing the egg or the sperm. So basically all of the egg cells that you get from a single female, they are expected to be different because of this process of meiosis. And all of the sperm cells that you get from a male, they are expected to be different because of the process of meiosis. If you need to go back and take a look at meiosis again, this would be chapter 13 in your textbook. So meiosis is one big contributor to the genetic variation we see from sexual reproduction. So that would be number one. We also have a lot of genetic variation. So let's see, that's number one. We also have a lot of genetic variation due to the fact that we're getting random fertilization. So this random fertilization has to do with the fact that it is going to be a random egg up here that is chosen from a female, and it is a random sperm that's being chosen from a male. And so they are randomly combining to give us some zygote. The end result of all of that is that you end up with a lot of unique individuals, and you could take the same two parents, you have the mom and dad, and if they reproduce a bunch of offspring, we still expect each one of those to be different. And if you think about some insects and other animals that produce a lot of offspring, we could be talking about thousands of offspring from the same two parents. So every single one of those will be different from each other at the DNA level. So that's one place that the genetic variation comes from. Some genetic variation also comes from the formation of new alleles. When we're talking about this, primarily we're talking about mutations. Okay, so mutation is the place where we really get our raw material for evolution. So new genes are formed in this way. So in this way, we're talking about a change in DNA sequence. It is going to be a random change in DNA sequence. And so that random change may be something that ends up being good. It may be something that ends up being bad, but it is going to give you some new combination of nucleotides that natural selection could actually operate on and work on. So if we look at this, here we have an example that you looked at in Bio 1. We have the hemoglobin gene. This gene is responsible for sickle cell anemia. In the normal, which we call wild type, so wild type is normal, and someone that does not have sickle cell, this would be the DNA sequence that we see, CTT. That gets transcribed into a piece of RNA, which has GAA in that same location, and then that tells the cell how to make the hemoglobin protein and the specific amino acids that should be there. This mutation right here, where you get an A instead of the T that we had over here, that is what ultimately causes sickle cell anemia. The difference is that we get a different RNA molecule, and then the RNA is what the cell is going to be actually reading so that it can make the hemoglobin. And so the template that is being given to the ribosome to actually read is going to be different. And so these proteins, they don't fold the same. They end up doing different things inside the cell, and the one ends up leading to sickle cell. So this is an example of a mutation that turns out bad. You could also have a mutation that gives you better function. However, keep in mind that most mutations are going to have no effect, or they're going to be detrimental. It's really very rare that you have a mutation that provides an advantage to an organism. And the main reason for this is that there has been so many years, billions of years, that this process of evolution has been in the works. And so over time, basically things have been worked out really well. So evolution and natural selection together have produced the proteins and the gene sequences that make us up, and those are ones that work together really nicely. A third place where we get genetic variation is altering gene number and position. So sometimes when we have reproduction, you do have a gene that gets duplicated. You could also have whole sets of chromosomes that get duplicated, and this could cause a change in the phenotype of an organism. So in this picture here, we have an example where you have basically a duplication of the chromosomes that's taken place. So when you look at those, they do look a little bit different, the two flowers, and it's due to them having a different number of genes. So one has twice as many as the first one does. And the last place we're going to get a lot of genetic variation is from rapid reproduction. Rapid reproduction basically leads to, this is going to lead to basically fast evolution. And so more generation rates. We're talking about generation rate in humans. We're looking at 15 to 20 years, really. If you're talking about some animals, it may be 6 months, it may be a year. If we're talking about things like bacteria, we're talking about maybe 20 minutes. Viruses are another thing that has a very fast reproduction rate or very fast generation time. So this is why we hear about bacteria evolving so rapidly. So now they're resistant to new antibiotics that they weren't resistant to before. Or this is the reason why we have to continually update the flu vaccines or viral vaccines, because you do have a lot of changes that take place, and there's a lot of generations that evolution can act on those changes. It's just, I want to emphasize it again, it's extremely important that we understand that evolution occurs in populations, not in individual organisms. So if we talk about, if we're emphasizing this again and again, the fact that this occurs in a population, what do we mean by a population? So by a population, we're basically talking about interbreeding individuals. Okay, so these are individuals that would come into contact with each other enough so that you could have reproduction taking place. So they are going to be producing offspring with each other. Populations can be isolated by different methods, and we'll talk about that in later chapters. So the collection of individuals is what can evolve one direction or another, not each individual in that population. When we talk about the gene pool is basically all the alleles in the population. So if we're talking about all the alleles in the population, keep in mind that these are diploid organisms. Each individual is going to have 2 alleles, and so the gene pool is basically the sum of all of those. So if we had a population of 1,000 individuals, so looking at these pictures here, so if we've got 1,000 of these cattle here, each one of those, if we're talking about one gene, each one of those is going to have 2 alleles for it. So we would have a total of 2,000 alleles to dump into what we're calling the gene pool here. So gene pool, the collection of all the alleles in the population, which is the interbreeding individuals.